and welcome. In this tutor, we are going to discuss about the remaining structures of the cells like lysosomes. Lysosomes, lyso means splitting and soma mean bodies. It was discovered by Dida in 1949 first times. It is very important. Substances, the basic purpose is to digest the foreign substances that gain entry into the cells. Likewise, here is the cells. Any foreign substances that gain entry into cell is immediately engulfed by lysosomes, and it contains a hydrolytic enzyme, nearly 50 hydrolytic enzyme that can digest the foreign material into the particles, and they are most abundant in those animal cells which exhibit a phagocytic activity bounded by single membranes and the simple sac rich in acid phosphatases. The enzyme or the acid phosphatases, and these enzymes are synthesized by rough endoplasmic reticulum and further processed by Golgi vesicles. Enzymes are produced by endoplasmic reticulum, and its codes and its vesicles are bounded by Golgi vesicles, and it is converted into the final product in the Golgi vesicles. Processed enzymes and budded of Golgi vesicles are called primary lysosomes, and when primary lysosomes get involved with the food vacuoles, it is converted into secondary lysosome. Like here is our cell structures. Here how the lysosomes are produced. Rough endoplasmic reticulum produce the enzymes of a particular type of the lysosomes. Then it is converted to Golgi apparatus and finished product. Here is the lysosomes, primary lysosome. When a foreign particle is converted into the cells, it is packed inside the vesicles, and there is the formations of the transport vesicles, and when it enters into the cells, it is it is given to the lysosome, and lysosome attached with its, and the enzymes of lysosome enter into these food vesicles, and digestion starts. Its enzymes digest the food particles, the lysosome. Then the food digested product may be excreted out of the cell, exocytosis, or maybe reused. It is very important organelles. It destroy the foreign particle and also destroy the internal cell membranes, cell organelles that are no more usable. Like it is engulfed the old mitochondria, and this product and the product obtained by this way can be used to regenerate new mitochondria. So it is phagosomes and autophagolysis and autophagy. Autophagy is the eating process of its own materials, like starvations, like, or also in starvations, it can digest its own particles. In normal physiological process, the part of the cells are engulfed by primary lysosome and digested to generate energy. It also produces energy by digesting the internal cells or organism. And in autophagosomes, when it's part of its own, it is called autophagosome, and the process is called autophagy, self-eating process, and it is used in renewal of the cells. Then we will discuss about the different diseases caused by deformities of the lysosomes. There are the various kind of lysosomal enzymes that are breakdown of the various type of the materials. If these enzymes are present, these substances that have to be hydrolyzed by the lysosomes get accumulated in the cells, and this causes diseases like glycogenesis type 2 diseases, in which the enzymes that cause the glycogenolysis of diseases is absent, and so the glycogen is stored in excessive amounts. Yeah, like 20 such diseases are nowadays because of the absence of particular enzymes, and Thai such diseases is because of the enzyme that is involved in catabolism lipid, and that particular enzyme is absent in the Thai such diseases, and the lipid become to accumulate in the body, and accumulation of lipid in brain lead to the mental retardation and even death, and it is very dangerous circumstances for the body. And then there is no paroxysm. The vacuoles are the organelles, many small organelles found in the animal cells, and the one central large vacuole found in Plant cells, actually the plants at its early times have many small vacuoles, and the central vacuole is formed by coiled cells, and the combination of the various smaller vacuoles act as a storehouse of the various chemical, and it is also provide turgor pressure, it provide rigidity to the cells, and it is found in both animal and plant cells. And in plant cells it is large and expel the nucleus to the periphery, and it is bounded by single membranes. Like vacuoles are formed by the Golgi vesicles. Vacuoles serve to expand the plant part without diluting its cytoplasm. It expand the plant parts and give the rigidity to cell along with its cell wall. It gives the rigidity to the cells and maintain the shapes of cells. Also function site for the storage of water and the small product or the metabolic intermediates. Plant vacuole is contributed to turgors for individual plant cells contribute to the rigidity of cells, and a younger part of the plants would die and wilt if there is no turgor pressures. Then our next organelle is mitochondria. Mitochondria is very important organelle and powerhouse of cells. It gain energy from the food particle and convert it into the ATP. And we have discussed the role of the mitochondria in the cellular respiration, the bioenergetics, or how it produce ATP. It is very important process. It is like it is composed of double membrane. Outer membrane is smooth and inner membrane is folding cisternae. It is the site of various like oxidative phosphorylations and respiratory chains, and its folding is characteristic. The mitochondrial membranes are similar in structure to the other cell membranes. It is the similarities. It is the organelle which contains its own DNA as well as ribosome. It is also called semi-autonomous organelles because it's produce it contain DNA, so it's produce its own RNA and its own protein particles. So it is a self-replicating organelles, and it was produced by symbiosis mechanisms by engulfment of the certain type of the bacteria that are involved in cellular respirations. Along with the chloroplast, it is also a symbiotic organelle. Inner surface of the crystal of the mitochondria has small like knob-like structures called F1 particle. These are actually the ATPases, have help in the production of the ATPs. Mitochondrial matrix contain large number of enzymes, coenzyme, organic and inorganic salt, which it help in the vital metabolic processes like that mitochondria are involved with, like Krebs cycle occur in mitochondria, aerobic respiration, fatty acid metabolisms. As a result of these chemical reaction, energy is formed in the form of the ATP, and when ATP is provide energy to to the body, it is converted into ADP, and ADP is again converted into the ATP by the mitochondria. Here are the structures. Here is the outer smooth surface. Here is the inner forming cisternae, and it has the F1 particle, and its ether sides. It is the outer membranes. Then it is the outer compartment between inner and outer membrane. Then here is the outer compartment, outer membrane, inner membrane in the form of Christie, and then there is the inner compartment and matrix or matrix. Outer compartment, inner compartment, outer membrane, inner membrane, F1 particles, cisternae. The most important MCQs here, the Christie is made up of the lipoprotein membrane containing different enzyme as well F1 particle embedded in it. After a specific process in the inner mitochondrial membrane is ruptured and F1 particles come out on the surface. Certain type of processes, the inner mitochondrial membrane is ruptured and it become at a surface, and Christie are made up of the lipoprotein. It is very important. Here is the how ATP is produced by the mitochondria. The same lines, plasters of the only found in plant cells and help in the photosynthesis, and pigment containing organelles. Plastids are classified into chloroplast, chromoplast, leucoplast, amyloplast, proteinoplast, according to various structures. Chloroplast are you know about the photosynthesis, and you know the structure of the basic structure of the chloroplast. Chloroplast is also the double membranous organelles. they contain green pigment, and green pigmented chlorophylls. It is the same like as like the chlorophyll has the same structure like hemoglobin, except to contain magnesium instead of irons. Chlorophyll molecules resemble hemoglobin of the hemoglobin, like is same difference is that it contains magnesiums. Then chloroplasts vary in the size and shape with 4 to 6 micrometer. It is important structures under light microscope. It can structures composed of structure like grana embedded in matrix. Container and matrix are the two important structures seen under light microscopes or under electron microscopes. It is composed of three layers like envelopes, stroma, and thylakoids. Envelope is the double membranes, while the stroma cover most of the volume of the chloroplast. It is the fluid, and it contains the protein. Stroma, you know, the stroma is the process where light reaction produce and sugar are produced. This part of chlorophyll CO2 is fixed because fixed in dark reactions, and dark reaction takes place in stroma, and the photosynthetic process takes place at grana. Some proteins are also synthesized in this spot because it is also a self-replicating organism. It contains DNA of its own. The thylakoids are flattened like thylakoids. The thylakoid arranged in the form of grana. The thylakoid are piled on one on each other's like coins. There are 50 or the more thylakoid piled to form one grana, and grana are interconnected with each other by non-green part called as intergrana. Membrane of grana at the site where the sunlight energy is trapped. The photo light reaction take place at the grana, and dark reaction takes place in stroma. Like chromoplast, as the colored plastids present in the petal of flowers, ripen fruit, help in pollination and dispersal of seeds. Leucoplast are the colorless triangular underground part of the plant cell, help in storage of food. Here are the structures of the chloroplast: outer membrane, inner membrane. Outer and inner membrane both are smooth. Here the difference between chloroplast and mitochondria is that here the both the membrane are smooth. Here are the thylakoid pile in the form of the grana, and thylakoid interior space is called lumen, while the interior space of the chloroplast is called stroma. We know well. Then they are connected with each other by green parts. Here it is called intergrana, which hold this nucleus is the central part of the name. But you know, it is very important how it was discovered. It was discovered 1831 by Robert Browns. It was very dominant because it was a darker space near circus. Nucleus may be irregular, spherical in shape, generally have one nucleus called mononucleates, but containing two nuclei or binucleate, and multiple nuclei can also be formed. Like in the muscle cell there are the multiple nuclei, like in the paramecium there are the two nuclei. Nucleus is the only visible when cell is in non-dividing state. When dividing, there is the dislocations of the nucleus is broken down. Nuclear membrane is broken down. The chromatin is converted into chromosomes. You know very well. Chromatin networks and soluble protein nucleoplasm, like cytoplasm. Here contain nucleoplasms. In dividing cell, nucleus disappears and chromatin material is replaced by chromosomes. Hereditary material is in the form of chromosome. You know very well. Like DNA, RNA, protein, enzymes form the chemical composition of the nucleus. It is very important. Nucleus consists of nuclear membrane, nuclei, nuclear plasm, and chromosome. We will study one by one. Nuclear membrane is outermost boundary. It is also a double membrane. Outer membrane continues with the endoplasmic reticulums are near the Golgi apparatus. Or inner membranes is inside of the cell and cover most of the inner structure and nucleoplasm and nucleoli, and there are the various spores in the cell nuclear membrane where the particles can enter into the nucleus, and exchange of material between the nucleus and the cytoplasm takes place. And nuclear pores allow the exchange of material, and at nuclear pores both the outer and inner membrane are continuous with each other, like yeah. They are continuous with each other at a nuclear membrane here. These are the nuclear membrane, and these outer and inner membrane are continuous with each other. A number of the nuclear pores may be variables like undifferentiated cells, large cells like egg cells. It contain be 30,000 per nucleus, or it undifferentiated cell like erythrocytes. There may be 3 and 4 pores per nuclei. There is no need of much pores because of there is no DNA at mature erythrocytes. And nucleolus is the darkly stained body inside the cells, and it is the basic building and basic machinery for the production of the ribosome. So it contains ribosomal synthesizing material, ribosomal RNA, and there may be one or the more nuclei in the cells. It's composed of two regions. The peripheral region is a granular area. It contains ribosomal subunits, and central area is the fibrillar. It contains RNA and recombinant DNA or ribosomal DNA. It is the nucleolus where ribosomes are assembled and exported to the cytoplasm via the nuclear pores. Ribosomes are prepared in the nucleolus and exported outside to perform various functions. Chromosomes are formed by the chromatin condensed in the form of teams in the form of fibrils, and there are maybe two arms of the chromosomes, and they are attached at the centromeres, and there are the kinetochore protein at the centromere, and where kinetochore fibers during the cell divisions are attached, and it is the basics inherited. It is the unit of inheritance. Genes are found on the DNA that are in chromosomes, and chromosome is composed of DNA and proteins. All informations to control the activity of cell is located in chromosomes in the form of genes. A number of the chromosomes in particular type of species remain constant, like 46 chromosomes in a man, 26 chromosome in a frog. Chimpanzee there are 48 chromosomes. There are 8 chromosomes in fruit fly Drosophila, 16 chromosomes in onion, 48 chromosomes again in potatoes, 14 chromosome in garden tree. Number is in of chromosome in the germ cell is half as compared to somatic cells, like a body. In the body other cells there is diploid number of chromosome, where in the some germ cell there is the haploid number of chromosome, like sperm and egg contain 23 chromosome. Drosophila's like sperms and eggs contain 4 chromosomes, and in the same way here are the basic structure of the chromosomes. Here is the kinetochore regions. Kinetochores, sister chromatids. Both of the sister chromatids are exact replica of each other's. They are exact replica. Here is the new process of the nucleus. Here outer membrane and inner membrane. There is the nuclear pore. There is nucleolus, nuclear envelope, nuclear plasm, chromatin network, and prokaryotes and eukaryotic cells. The different biologists have divided into two types. Prokaryotes are with not definite nucleus, and eukaryotes have definite nucleus. Then nuclear material DNA is in line DNA. It contain of the eukaryotic so is enclosed in the nucleus, while the DNA of prokaryotes is submerged in cytoplasms. And diagrams are also different. Prokaryotic cells generally lack many of the membranous boundary structure as found in eukaryotic cells, like mitochondria, endoplasmic reticulum, chloroplast, and Golgi apparatus are absent in prokaryotes but present in eukaryotes. It is also a very important difference between prokaryotes and eukaryotes. Prokaryotic cell has no distinct nucleus, and its DNA is suspended in cytoplasm. The difference ribosome is found in both prokaryotic and eukaryotics, but the ribosomes of prokaryotic cell is 70s units, and that of eukaryotic cells is 80s subunits, and the ribosomes that is found in the mitochondria and chloroplast are also 70s. Because of the simpler structure is prokaryote, and eukaryote has complex structures. Then the most distinctive feature of the prokaryote is cell wall. It is also a very important MCQs. It is composed of polysaccharide chains bounded covalently to shorter chain of amino acids. It is called peptidoglycans and murines. An entire cell wall is regarded as cellular complex. It is called succulus. It is also a very important MCQ. The cell wall of plant is generally made up of cellulose and different from that of bacterium, and bacterium is composed of peptidoglycan. Here are the different structures found in a prokaryote. Generalized prokaryotic cells, it is a nuclei containing DNA, ribosomes, smaller ribosome, flagellum, cytoplasm, pili. Its flagellum composed of flagellin proteins. Why is the flagellum present in a particular type of eukaryotic cell like animal cells? It is of composed of tubulin proteins. There is cytoplasm. Here are the pili. Etc. are found. The capsule, plasmids, plasma membrane, cell wall, capsule, nucleoid, ribosomes are formed. There are also difference in the replication process of both prokaryotic and eukaryotics, and during the translations, the RNA is directly in the cytoplasms, while there is very post translational and post transcriptional modification found in a eukaryotic cell. Here we are end with our lectures.